Good afternoon, and welcome to Core Technology Conversations, brought to you by the Research Course in the Division of Research at Florida Atlantic University. My name is Thomas Graziano, and I'm the Director of the Research Course, and I will be your moderator for today. Research cores are federally compliant, fee-for-service, shared resources that provide access to instruments, technologies, as well as expert consultation and other services to scientific and clinical investigators. We currently have 7 active cores at FAU: the Biostatistics Collaborative Core, the Cell Imaging Core, the Clinical Research Unit, Comparative Medicine, the Engineering and Technology Core, the Neurobehavior Core, and the Water Analysis Lab. This seminar series will let you know more about those cores and give you a glimpse into some of the amazing research each one is capable of. Today's presentation will be presented by Jordon Beckler. Jordon currently serves as the manager of the FAU Water Analysis Lab Core and is an assistant research professor for the Harbor Branch Oceanographic Institute and the Institute for Sensing and Embedded Network Systems Engineering, also known as the iSense pillar. With that, it is my pleasure to introduce Jordon Beckler.
All right, well, I appreciate the introduction. Today, I'll just give an overview of the core as it currently stands, and then also some future potential expansion plans. I'm sort of going to give a large overview of all potential things that we could include. What actually ends up being included in the future depends on a lot of things like costs and, what's right for the Institute and whatnot. So we'll just go from there. So we recognize in the water analysis lab that Southeast Florida, and it's not only in the lab, but FAU also recognizes all the wonderful educational and research opportunities. We're obviously a regional leader. Florida is subject to all kinds of issues with water, soil, and sediment quality. There's other matrices, industrial waste, septic tank waste, and a lot of different faculty at FAU require these analyses in support of their research, but then we also have a growing external customer base. We even get referrals from manufacturers of the actual instruments that we use for our analyses. And obviously we are continuously trying to expand our capabilities, as you can see from the timeline back when Tara Root and the geosciences, or formerly of the geosciences department, established the core with the hydrogeology lab in 2013. Coastal studies lab joined in 2016. Then my own lab joined in 2019. And then after Tara left last year to take a different position, I then became the manager. And so we offer quite a few advantages actually over going with, let's say, a strictly outside analytical lab. Our analyses are very high quality. I think, obviously I'm biased, but I think that's absolutely the case. We have a dedicated technician who runs at least all the water quality work at Harbor Branch. So we have a very strict QA, QC procedure. We'll do a lot of extra steps in our facility. And a lot of this stems from the fact that we're not only the people doing the analysis, but we're also researchers. So we understand a lot of different constraints that come with a lot of different analyses. And then we're also able to provide the end users or the people that are contracting us, we're able to provide them with a lot of advice as well. And just some fun facts, according to Kermit, is that we are the first FAU research core with multi-unit managers. We're one of the few cores that's effectively able to cover some of our own costs with external customers. And we don't really have a dress code. We're pretty relaxed, minus the personal protective equipment. So we'd like to think we're very approachable. As far as the core facility folks, probably the most important person is Lynn Wilking. And that's at least important with respect to our lab at Harbor Branch. She basically runs all the samples and coordinates the intake. Down in Tiffany's lab, Nick Brown tends to do a lot of the research. And then finally, of course, there's me. And I, of course, have to thank Gabe and Thomas. They have offered awesome assistance with the management of the core. It takes a lot of, the stress off of me. So what we currently offer is in black. So a lot of basic water quality analysis, almost everything you could think of, we have a way to do. There's a couple of items that the status is a bit uncertain since Tara left, but eventually we can probably get these things going again. So of course, you know, we're running nutrient analyses, major ions. There's some things that are a little more obscure, you know, hard to categorize: equipment rentals, instrument use, training, etc. And then finally, the things in blue, these are services. Notice they all have, or most of them have, a common theme related to sediments. And that's sort of the avenue that I'd like to expand into. So along with that expansion may come change of the name. So it wouldn't strictly just be the water analysis lab anymore. Just a few examples of some of the instruments that we have. The SEAL nutrient autoanalyzer is something that my lab inherited when Tara left. And this is really a mainstay for analysis. It's probably the most commonly used instrument in the world for nutrients. So all sorts of research is supported in terms of ecosystem health, restoration of natural ecosystems, looking at algae bloom nutrient dynamics or aquaculture, the role of nutrients. And finally, of course, the fate of anthropogenic contaminants. This is my baby. It was the most exciting thing that I think I purchased since I've been here. This Shimadzu total organic carbon analyzer. It measures obviously total carbon, but then also total nitrogen. And so with those two things, you know, you can, and with these two things combined with the autoanalyzer from the last lab, it can really cover all your main nutrient bases. That's, you know, that's probably 90% of the analyses that people would want to do. We can also pre-filter to characterize the dissolved fractions, so dissolved organic carbon, total dissolved nitrogen. And really cool, unique advantage is we can add little stir bars to our autosampler vials, and there's a built-in stir. This is an extra feature that will then stir the samples prior to them being introduced, which allows you to measure particulates. So, you know, because of that, we could measure things like microplastics, colloidal fertilizers and feeds, and then, which is really important up here, and this is something we're really expanding upon. You know, we have a great optics group up at Harbor Branch, and this instrument can really help out with that. We also have modular spectrophotometric systems, and we can kind of plug and play using these fiber optic cables. We have, they're not all pictured here, but we have quite a few different spectrometers, light sources, and detection cells, so cuvettes or liquid waveguide capillary cells, so we can get a 0 to 28 centimeter path length, or perhaps even longer. So if you're familiar with spectrophotometry, it's basically 28 times the sensitivity that you would get with a 1 centimeter. Also configure the devices to allow us to do fluorescence and reflect instruments in addition to spectrophotometry. So right now, we're currently using these for trace level ocean analysis for looking at iron redox dynamics, and really with anything where you have volumes that are limited to where you can't use the SEAL autoanalyzer. So for us, sediment pore waters, that happens to be a limitation quite a bit. And then down in Dr. Brake's lab, they have a soil sieve. It's a Rotap sieve shaker, and it actually allows you to separate your soils into various size fractions, which are then weighed afterwards. And this gives you a size distribution chart, which is really important, especially in her realm of physical dynamic modeling of beaches and coastlines and whatnot. It also informs groundwater prediction. In my realm, habitat characterization, for instance, of seagrasses and whatnot. And then even more in my realm, I guess, is nutrient fluxes. So smaller grain sizes tend to end up or tend to generate larger sediment-derived nutrient fluxes, which are important from an ecosystem perspective. So this is the Harbor Branch lab. I don't have a picture of Tiffany's lab, I apologize, but our lab is right here. It's nice and sunny and bright. It's a really awesome place to work. My My office is actually right here on the other side, so I think I have the best view at Harbor Branch. I look right down the channel. It's really great. We have, you know, a water system, and it's actually off camera here, but we can generate large quantities of 18.3 mega ohm water, so that allows us to be pretty efficient with our analyses. We have a laminar flow hood that we can use if we're trying to do trace metal work or trace nutrient work. And then this is something that you don't see in a typical research grade geochemical lab. They usually tend to be pretty messy and full of mud, imagine that. But you know, we developed an inventory management system, and we got this really nice, expensive label maker, and all of our samples that are introduced into our lab all end up with an ID number, and we can scan them when we're trying to do work on them, and all of that gets logged. It's a really great system, and there's a lot of work to set up, but it's definitely worth it now. And it ensures that, you know, we won't mix up samples when it comes time to analyze them. So I don't think we've made a big mistake yet. So some examples of the research that we can enable. So our largest external customer is a lab at the Woods Hole Oceanographic Institute. And specifically, they are looking at these Kettleponds up in Cape Cod. And these Kettleponds they're like these little confined, vertically stratified ecosystems. So you don't really get much vertical mixing, especially over the summertime. But they do have a very interesting array of microorganisms. And what they're looking at specifically is how the parasites then affect these microorganisms. And in doing so, these cells, they will basically lyse, which means they are destroyed, and you end up releasing the dissolved organic carbon, the dissolved organic nitrogen, particulates, etc. And so these are all the parameters. I mean, this is basically the entire point of their project, is to characterize the cycling of the nutrients that we are measuring for them. As far as an internal project, this is one that we're involved with. It's the integrated multi-trophic aquaculture system. We are growing, well, myself and many other collaborators are growing halophyte, salt-tolerant plants for human consumption. And the waste from fish aquaculture, the waste products actually go in to fertilize these plants. And so specifically, what my interests are to look at the choice of how the substrate, whether these plants are rooted in sand or clay pebbles or just grown purely hydroponically, how does that affect not only just the growth of the plants themselves, but then in turn, how are those effects, well, are they caused by differences in nutrients? And we actually have some pretty cool preliminary results right now showing that indeed you get better results, better yields from sand, because it seems like the sands, well, let's say within the sands, you have a says that you have nutrient uptake, which implies that you need a nice, healthy, sandy root environment to have a nice microbial community that's going to work in sync with the plant to actually create the nutrients in a bioavailable form. So now this work is not funded by the Core, but to be honest, I don't have that many examples of work that, or results from work that has been funded by the Core, because we don't really see, you know, we just provide data. We don't really see the actual results in action. We don't see the post product. But this is something, a project that I have that is exciting and also represents a lot of the different analyses and incorporates a lot of the different analyses that we would offer, or that we do offer with the Core. And so these blue holes are all over the Western Gulf of Mexico. People have no idea that they're even there. Basically, it's a hole in the bedrock. This one happens to go down 400 feet almost. And we're trying to understand quite a few things. If there's flow coming from these blue holes, what causes the fish to aggregate there. These blue holes are definitely known as hotspots, as ecological hotspots. You could just see how stark the transition with the bedrock really is. And you end up with an anoxic environment. So basically there's no oxygen once you get into the hole. It all ends up being extremely sulfidic, which makes for a really cool geochemical environment. Just gonna play a video of what we found down there. It doesn't really have much to do with the core, but you know, it's certainly cool, worth watching. This is a sawfish that must have swam down there. There was actually 2 of them. They swam down there. They suffocated due to sulfide toxicity, and then they were preserved on the bottom because there's no oxygen. They're essentially preserved under those sulfidic conditions. So when they were retrieved by the Fish and Wildlife Commission, they said it was the worst smelling thing they had ever dealt with, as you can imagine. We have no idea how long they were down there for. You re-maximize, and so these are some of the results. We present this data as profiles, right? So here is a depth profile, and then on the y-axis here, on the x-axis, usually we think of them as being reversed, but the y-axis is concentration now. So you can see if you follow your oxygen, it's saturated above the blue hole, but then right when you get into the blue hole, it basically goes to 0, right? There's something going on here we're not sure of, but you end up with these very sulfidic conditions down on the bottom. And sure enough, you see hydrogen sulfide in green right down here, or I'm sorry, hydrogen sulfide is in blue. And then you have all this sulfur cycling, these different redox states of sulfur. And this high redox, this high geochemical activity is actually associated with enhanced nutrient cycling. So indeed, we have a lot of phosphate that's coming out of these sediments, apparently. We also have a lot of ammonium. So both phosphorus and nitrogen that are coming out of the sediments, and they're being transformed so that ammonium is being converted into nitrate then. So it's like you have this whole little microcosm of biogeochemistry occurring in this blue hole. And the core measurements allow us to decipher this. So it's like a big puzzle. And what are some implications? Well, these blue holes are basically what we call ecological engines because we haven't come up with a better term for them, but they aggregate fish, right? They're shelter. Then they end up like a funnel, depositing all kinds of organic carbon and nutrients. It just sort of filters down into the blue hole. It collects in the bottom of a debris pile, but then those nutrients are converted by bacteria back to their inorganic form. And then those flux upwards. Those flux upwards, they stimulate phytoplankton. Those phytoplankton are then eaten by fish and shrimp and whatnot. And you actually end up, or phytoplankton are eaten by zooplankton, which are eaten by fish. And you end up with this ecosystem that's sort of self-sustaining and can expand laterally. So these blue holes are really an origin of life out on an otherwise bland Gulf of Mexico continental shelf. They're also the only, to our knowledge, the only permanently stratified marine, entirely marine basin in the Western Hemisphere. You have to go to the Black Sea or the Baltic Sea to find anything else like this. I don't have data for it, or I do have the data, but I'm not going to talk about it. But just to excite everybody, we did document on our last trip exchange with the subterranean aquifer, with the submarine aquifer 50 miles offshore. Was a different blue hole, but happy to talk with anybody afterwards that's interested. So as far as the new directions that I want to take the core, I focus on sediments, and sediments are really relevant to the world right now. And I think more and more people are learning how important sediments are as we go on day by day. And you can see how diverse different sediments are, right? You have what we call early diagenesis. That's when carbon and nutrients and things end up in the sediments and they cause changes over time. And so the deeper you go, typically the older the sediments are. Now in shallow water bodies like the Indian River Lagoon or Lake Okeechobee, you can have a lot of exchange. If there's nutrients coming from the sediments, you could imagine they would make a disproportionate impact in those shallow basins. Sediments are extremely important in terms of value, right? The dollar value. So the most important ecosystems on the planet, besides coral reefs, are coastal wetlands, freshwater wetlands. And of all the main value that humans derive from them, waste treatment is the most important. So anything coming off of land, any excess nutrients that would cause eutrophication and hypoxia in the ocean and algae blooms, they tend to get taken care of quite a bit by wetlands. So you can actually assign a value for what that's worth. And if you actually look at the sustainable annual value per hectare, that's 2.5 acres. Basically, in 2011, these values changed over time just because we recognized more ecosystem services as they emerged, as our scientific understanding improved. But we're talking 250 grand a year from tidal marshes and mangroves. And so it just goes to show that sediments are extremely important, and there's a lot of money surrounding them, potentially as income sources for the Corps. You know, there's justification in working on sediments if they're worth that much money. So I'm going to focus specifically now on an example for algae blooms. So in Florida, right, we have a ton of algae blooms. We're perhaps the worst in the world, although they are increasing in globally. Well, oh, sorry, I don't mean world. China is the worst in the world, but Florida is the worst in the US. A typical sort of paradigm is you'd have nutrients, nitrogen and phosphorus coming in, let's say from agricultural or primarily natural sources, but also agricultural septics, et cetera. Those nutrients, they end up sedimenting out, right? Or they're incorporated into plankton, which eventually die and they're settled out. And they would otherwise sediment out and be stored in the sediments. You have, in a lot of cases, a lot of iron oxides, and they're like sponges for phosphorus. So they store this phosphorus, they can sequester it, and it won't, you know, cause algae blooms until, under certain circumstances, if the sediments are hypoxic, or if the water column's hypoxic enough and the sediments become anoxic enough, you can release that phosphorus. You'll dissolve those iron oxides and you release that phosphorus. We call that process internal loading. So you have external loading and internal loading of nutrients. And the relative role between external and internal loading are extremely important to characterize. Eventually, any nutrients can also make their way out to the coast and cause red tide. Now, it's easy to measure the nutrients going out, coming in, and in the water column. We do that all the time. There's in situ sensors that can do that for us. I shouldn't say easy, but it's, you know, straightforward, I guess. It tends to be expensive, but you can do it. On the other hand, measuring the fluxes, measuring this internal loading is not easy, but this happens to be where our lab specializes. And so I think this would be a great addition to the core. The main importance, right, is if internal loading is important, like it is in Lake Okeechobee, for instance, you could stop the inputs from agricultural sources from the rivers tomorrow, but you'd still have this huge load, these legacy loads that continue for decades. And so as an example of some of these techniques that we use, I'm going to present you data from our new project. It's called the HALO project. And we're trying to understand what's causing algae blooms in Lake Okeechobee. We have a lot going on that I won't explain everything in detail, but task 3 is my stuff. And we are collecting sediment cores, right? So we actually collect samples of sediments and we measure the nutrient flux directly from those sediment cores. And then we also deploy in situ instrumentation, not this exactly, but similar to this, that allow us to these nutrient fluxes from sediments directly without having to actually take samples. And so as far as a potential future water analysis lab service, something that's kind of obvious, it's sort of on the fence how you would even monetize something like this, but it is a skill, and I think it's a concrete skill, is we have a lot of experience in designing field sampling surveys. You have to know your goal, and you have to consider and balance a lot of different variables. And really, you know, the best solution only comes with lots of different experience, with lots of experience. And that's what we have, you know, between Tiffany and myself. You know, we have a lot of combined experience working in everything from lakes to beaches to deep sea hydrothermal events. So we can really ensure that if we're designing a sampling routine, it's going to ensure realistic expectations. So in this case, we need to collect sediment cores. Right now, the core does not, or the WAL Core does not, this is not a service. Sediment sampling is not something that we can currently charge people for, but sediment sampling is not straightforward. It's something we have a lot of experience in. If you want to collect undisturbed sediment cores for whatever reason to measure fluxes or to measure contaminants, it's something that you need to know how to do. And we have quite a few techniques for that. We can also use something called a ponar if we're just trying to collect grabs of sediment. For Lake Okeechobee, we actually contract divers, but we do have an arrangement for that. So that is something we can also do for external customers. This is probably the most unique offering that we would have. There's probably only a half dozen labs in the country, maybe the world, that do this. This is called electrochemical profiling. What we have are these electrodes that we make ourselves and a microprofiler. So you can see your sediment core here. We can put an electrode here, and this electrode will slowly profile downward. Over the course of a few hours, this black manifold here will move downward. And at each downward step, on the order of a millimeter or so, we run a series of scans at this electrode. And these series of scans allow us to measure a lot of different redox analytes that are involved in sediment nutrient cycling. So not only can we measure the nutrient fluxes itself, but we can figure out why you have a nutrient flux. So for instance, if it is because you have iron oxides that are dissolving and releasing that phosphorus, we can detect that. And that's exactly what we detect right here. But we can't measure everything using those techniques. So we do have to also process sediment cores. And this is something that the Harbor Branch engineers actually designed for us recently. And it's awesome. It's so awesome that we're actually going to publish it. We just put in a paper. It is this is a device, it's basically an improvement on existing devices that allow us to process a sediment core. Notice you would have a core here, you have this piston, and you push up this piston using a jack bit by bit, and you get centimeter by centimeter, the mud comes up, and we can collect that sediment, put it in a test tube, and then that becomes our sample. We also can easily attach a glove bag. This is the most unique part right here. So we can attach a glove bag and then fill it with nitrogen. And then this way we won't have any of that nasty oxygen in the atmosphere interfering with our redox chemical analysis. But again, it's an efficient way to process sediment cores. And when we do process sediment cores, what we do, this is pretty typical of it. So this is from the Lake Okeechobee project. These are the first samples that we collected back in mid-December, first of many. And you can see we can analyze the solid phase, like we can directly measure those iron hydroxides and the phosphate that's bound to them. And you can see those two sort of co-vary, the green along with the bar. But then we can also measure what's in the pore water. So notice here you have ammonium increasing with depth from this sediment core. Whereas over here, you don't seem to have as much, right? You don't have as much, maybe what, 100 micromolar or so. And so what we can do with that is we can take these gradients. We will take this gradient, this change in concentration as a function of height right here. And we can just multiply that by a couple of items, and you end up getting your flux. And your flux is in the units of millimole per day per meter squared. So essentially, think about this like kilograms per meter squared. So that's an estimate of the flux from this exact location. And we can then extrapolate those fluxes because we know the area, this is the surface area of the sediment that flux applies to. So we can expand that to get a lake-wide estimate. And notice we did take, 3 or 4 cores here. And you can see, of course, at the north core, you get a much higher ammonium flux than you do here at this east core. Here's north, here's east. So you catch my drift. Now, one more thing though, is that these fluxes are not, these diffusive fluxes that we get from these cores, they're not a perfect measurement. When you take a core, you're actually perturbing the sediments. You're removing it from its natural environment. You're eliminating any physical processes that might be enhancing or accelerating the flux of nutrients. And so it ends up being much more valuable if you can perform this work in situ. And so what I mean by that is finally this really cool new Benthic lander that we developed specifically for the Lake Okeechobee project. And I'll show a little bit how it works. PBS made a
Birds can only stay at a depth of 300 feet for a short period of time, whereas the lander can continuously collect data over a 24-hour period. One component, called a benthic flux chamber, samples nutrients, carbon, and oxygen that transfer or flux in and out of the sediment.
Think about like taking a coffee can and inverting it on top of the sediments. Now you have a good little volume of water, and we have syringes that take samples from that little parcel of water every 4 hours, and they're sampling both inside the chamber and outside the chamber. Outside the chamber is sort of a reference, like a background. Inside the chamber tells us how much is actually coming from the sediments themselves. So that's how it works. It's really cool that PBS actually made a documentary about the blue holes. This isn't the same lander, as you can tell, but it's a similar idea. It's a great way to, it's a great visual, right? It's kind of hard to explain that to people, but thank God PBS made that. So here are those two benthic flux chambers we have on this lander, but then we also have what I showed you before with that electrochemical profiling system. We have an in situ version on that. So we'll deploy this lander overnight at a site. We will get an estimate of that flux. And finally, we will also get an electrochemical profile like I showed. So not only do we get a flux, but we also get a context for the flux, right? We understand why there is that flux. Here's a picture of us deploying it back in mid-December as sort of our test deployment in Lake Okeechobee. Here's just an example of the data and how we treat the data. Here's the time after the lander or after the benthic chamber essentially seals. Normally this would be about 24 hours, but we were just doing a quick systems test. And you measure the nutrients. So you could see the nutrients, they would theoretically increase with time. And you could take that increase and you could normalize it to the cylindrical surface area of that benthic chamber. By doing that, you could again come up with a flux in units of, let's say, grams per meter square per day. You can extrapolate that over the area of the lake. Now, I didn't do it here because something fishy happens here, so I don't trust it. But anyways, we've had quite a few different inquiries because of the media attention that we've had with this from external agencies that want to basically reproduce this lander. And I don't think they realize that how much goes into it. So it does seem like a service that we could potentially provide, although it wouldn't be cheap. And you know, there's one more thing that I'm not going to show, but we are now deploying electrochemical sensors, not just to collect a single sediment core profile like this, but we're now deploying sensors long-term so we can monitor how the sediment geochemistry changes over time. So like when you may have a nutrient flux, when you would have your oxygen be consumed rapidly, when you would dissolve those minerals, and then therefore when you would release that phosphate. We're trying to catch that in real time one day. But anyways, this is the end of the talk. I just want to sort of wrap up and say that you know, we obviously have a convenient multi-campus presence. Measurements for existing analytes. We can almost entirely characterize nutrients in any sort of natural system. We are ready and we're engaged, and you know, we're informed on a lot of different regional and global issues. And our future services can be anything from routine. You know, one of those sediment core nutrient analysis could be on the order of, let's say, $2,000, just a sheer guess, right? But just to give you an estimate, something like a benthic lander deployment would be a lot more expensive. We obviously have, you know, not just my own lab costs, but there's institute costs that are incurred as well. So, you know, there's a lot to think about here before we would ever be able to do something like that and start offering a service like that. But, you know, if you're interested in obviously us doing work for you or potentially providing a service to the Corps, contact me or Tom or Gabe. And I just want to thank, obviously, Tom and Gabe one more time, and Dr. Root, who's probably not here, but just thank her for getting this whole thing going. And finally, WHOI, Harbor Branch Facilities and Engineering for help with some of the work that you saw here. And of course, the sponsors of some of the research. And that's all that I have. So thank you. I'll stop sharing.
Great. Thank you, Jordan. That was an excellent presentation. It was really informative for sure. You have a lot of interesting research going on. Definitely. It looks very cool.
Cool.
Just wanted to remind all the attendees, if you hover your mouse over the Q&A button at the bottom, you can go ahead and type in any questions you may have. And let me see what we got going on here right now. Here we go. You mentioned that some projects were or were not funded by the Corps. Does that mean the Corps has support available for project needs?
No, the Corps does not have support available. If you're asking if we're receiving funding from the Corps, from FAU or outside, or from institute sources to do research, no, the answer to that is no. So any research that you saw here was either funded through external funding agencies or by paying customers, I guess. I hope that answers the question.
Yeah, I think you did. If by chance you have another question, whoever asked that, please feel free and type it in. Next one, will your Blue Hole project use the analysis available by the Water Analysis Lab for future samples, possibly to confirm field equipment results?
Well, we only have one more trip scheduled for May. And, it's kind of the question as to whether or not, if I'm doing my own research, does it make more sense to run it through the Corps, especially with a project that's already existing, right? To where we didn't initially budget that way. So from now on, I think the math works out better for me if I do budget. If I'm going to do research in the future, if I budget analyses through the Corps, even for my own research, I think that's still beneficial. So probably for the Blue Hole project, no. But in terms of confirming field equipment results, yeah, we are definitely, we use the, it's overlapping instrumentation. So we essentially use that instrument. That's kind of the whole point of the Corps, right? Is that we can use the instrument for research, whether or not it goes through the Corps or not, whether or not the funds move through the Corps or not. And so at the end of the day, we're still doing the same analyses, I guess. If that makes sense.
Yeah. Let's see what we got going on. Here's another one. You mentioned several future offerings. Can you give an idea of when they may become available? And how do we keep up to date, or how do you keep up to date with future offerings or new offerings?
Well, I bugged Thomas.
Yeah, I figured I was going to be part of the answer on this one for sure.
Yeah. Well, I bugged Thomas. There's obviously a lot of work that goes into defining the price structure, especially when it's something that no other lab in the world probably offers. And so, as far as a timeline, if I had to guess, probably the sediment flux work and whatnot, we would probably have that, let's say in the next year or so.
Oh, okay. Yeah, definitely. And we always
probably seems like a reasonable time frame. But these are items. These are things that I haven't even talked to you necessarily about yet, Thomas, or you nor Dave. So these are things that, you're seeing for the first time that I am, so.
Yeah, and whenever you're ready, we'll get that rolling as quickly as possible for sure, definitely.
Good deal.
And one last one, here we go. Can the Water Analysis Lab assist with preparing manuscripts after providing analysis?
Yeah, absolutely. It would definitely be, there has to be some sort of cutoff, right, as to where a reasonable amount of analysis, sort of a precursory analysis, I guess, comes with the data saying like, hey, look, the data makes sense or it doesn't make sense. But as far as involvement beyond that, I haven't had a situation yet where someone has really, really wanted us to be engaged. And so, I don't know when, personally, I wouldn't know when to say like, okay, well, this needs to be something that we need to then set up a new price for. We need to start charging for this data interpretation. But I feel like certainly for manuscript preparation, that would be something that, you know, there would have to be a separate agreement in place for that. That's a lot of work.
Yeah, definitely. And actually, that's all the questions at the moment. So I guess we're done there. If by chance anyone has any other questions after we're done here today, please feel free to reach out to Jordan directly, or myself, or Gabe, or research cores in general, and we'll help you with anything you need. So I'd just like to thank you, Jordan, for your presentation today. It was awesome. And thank you everyone for attending.
Great.
We're done.
All right. Thanks so much, Thomas. Appreciate it.
Thanks. Have a good one.